Oh, hello, my young friends. So today we're going to be talking about Unit 2, which is going to be cells and tissues. So we're going to look at cell structure. Now this should be reviewed, so you should know some of these. So, talking about a typical animal cell, going to be round, or at least not a certain shape, versus our plant cell. Our plant cell has a cell wall, so it's going to have a rigid shape. Now let's talk about the pieces. So first we have a cell wall, which is going to be found in plants. Plants, has an inflexible barrier providing structure, support, protection, and it allows molecules to pass through. Again, not in animals. Now we have the nucleus in both plants and animals, and this is the brain of the cell, contains the directions to make proteins by way of the DNA. The DNA is going to be your genetic material that tells and makes up you. Functions, it controls all portions of the cell. The nucleus again contains the chromatin, which is our genetic material, which is known as DNA, which is going to be deoxyribose nucleic acid. Also inside of our nucleus we contain a nucleolus, which is a dense part in the center. It'll typically be darker, and it is where ribosomes are created. And some cells have more than one nucleolus. Next we have the nuclear envelope. So the nucleus has this envelope around it that is a double membrane, and it allows proteins to pass through what is known as a nuclear pore, or a hole. Such as, if we look here, we see our double membrane, and then we see a pore, or a hole, in which things can pass in and out of the nucleus. If you can think of a wiffle ball that has multiple holes in it, that would be the shape of a nucleus. Next we have the cytoskeleton. It is composed of network of protein fibers. These protein fibers are typically about 3 of them. They go from the smallest to the largest, with microfilaments, intermediate filaments, microtubules, which are the largest. They are constantly changing position to help maintain the structure and shape of the cell. And the function is to provide structural support, and it aids in movement of the cell and within the cell. So again, we see a cross-section of the cell here. We see some certain organelles, but then we see all these little cross pieces. These are going to be our cytoskeleton. And again, we see the microtubule, which is the thickest. Then we have our intermediate filaments, and then we have our microfilaments, which are the smallest. Remember these, because you will see them again. Centrioles. Centrioles are non-membrane bound, so they're just structures that are sitting inside of the cell. There are 2, and they consist of hollow cylinders, or the microtubules. Remember, those are the largest types of cytoskeleton, or protein fibers that we will be talking about right now, that lie at right angles to each other. So, here we go. Each one of these yellows are going to be a microtubule. They help in the aid of cellular reproduction. So as a cell actually grows and divides, it will help keep everything where it's supposed to be, so that when it divides, it has equal amounts of different items. So these are not found in plants. The cytoplasm versus the cytosol. The cytoplasm is the area that consists between the cellular membrane and the nucleus. It is the whole area in between those two. May consist of organelles, may consist of the cytosol, which is the jelly substance, and it may consist of our cytoskeleton. But everything that lies within the plasma membrane or cell membrane and the nucleus is the cytoplasm. Now, again, that jelly-like substance that is in the cytoplasm that sort of holds all the organelles in place and holds everything where they're supposed to be is called the cytosol. It is a clear jelly-like substance in which all the organelles are suspended, aids in transport of material, and serves as a place for many chemical reactions to occur, especially for metabolism inside of our cells. Then we have our ribosomes. Our ribosomes are RNA, known as ribosomal RNA, or you might see it as rRNA. It is made in the nucleolus of the nucleus. There are 2 types. There are free-floating out in the cytosol, and then there are fixed. Some are actually bound to the rough endoplasmic reticulum. They do not have a membrane that surrounds them. They are made up of two subunits, a large and a small, and they're used for protein synthesis to make our proteins. So again, these are some that are attached here to the endoplasmic reticulum, and we have a small subunit and a large subunit that combine together to create the full ribosome. So the endoplasmic reticulum, that you will know as the ER, is another site for chemical reactions within the cell. There is a system of internal membranes, so they sort of look like a maze, and there's two types. There's the rough, that is speckled with ribosomes, which make it rough, and then there's the smooth, which is a series of the internal membranes that do not have ribosomes. The rough ER has a rough texture, studded with ribosomes, site of protein production. It produces proteins that are used outside of the cell, such as hormones. Now, smooth ER is smooth looking, and its functions are lipid synthesis, carbohydrate synthesis, the creation of glycogen, which is going to be where we store our energy, and drug detox for the cell. The Golgi apparatus is going to be our UPS, our shipping and packaging plant. It is membrane bound, and it functions to refine, modify, and package proteins to ship them out of the cell. So an example is we have our nucleus, and it sends out the RNA over here to the ribosomes on the rough ER to create the protein. Well, some proteins that just come out of the ER can go straight to the cell membrane and go out, but others must be packaged. So they come over here to the Golgi apparatus and go through a series of packaging processes. Then it is allowed out of the cell. Lysosomes, which are our garbage disposal units. They contain powerful enzymes that break down digested proteins, carbohydrates, and nucleic acids. They function in cleaning up the debris of cells, worn out parts, so that we could reuse the pieces. They destroy bacteria and viruses that are not supposed to be there, and they are for digestion within the cell. Now vacuoles. Vacuoles are sacs. They are membranous sacs, and they provide temporary storage for food, enzymes, waste, water, other needed materials. And there is a very, very large one in plants known as the central vacuole. It takes up most of the cell. Now, in animals, we do have vacuoles. We do. They're just not as large as the central vacuole in plants. Such as food vacuoles here in a paramecium. It holds the food once it is brought in. Chloroplasts, only found in plants. They are membrane bound. They contain circular DNA, and their function is to do photosynthesis, to create energy and food for the cells. Again, only in plants. So, here we see a 3D dimension of the chloroplast, and we have different pieces inside of them. But we are talking about humans this year, not plants. So, as you may recognize this, we are not going to go into detail. Again, we have pieces known as the thylakoid membrane that trap energy, the grana, which are stacks that help in photosynthesis, and then we have the chlorophyll, which make it green. Then we have the mitochondria that are in both plants and animals. Both. They are the powerhouse, the mighty, mighty mitochondria! Double membrane. We have an inner membrane, we have an outer membrane, and it is a jelly bean that has a maze inside of it. It contains its own DNA, its circular DNA, and it functions to convert and release energy as ATP from food through cellular respiration. Please take down that chemical reaction. You will need to know it, and we will go through it. Again, it is a jelly bean that has a maze inside of it, and each of the folds in the inner membrane is known as the cristae, and the fluid inside of that inner membrane is known as the matrix, and we go back and forth in and out of that inner membrane so that we can create energy. Now we have cilia and flagella. Cilia are cilia. They are short, hair-like projections from the cell, and they do a wave-like motion. We have these in our lining of our trachea or our windpipe, and it sweeps mucosa and dirt out of our lungs. And in females, yes, the females out there, you have cilia beating in your fallopian tubes. They move the egg every month from your ovaries to the uterus. Now flagella. It is a tail-like projection much longer than the cilia, and it does a whip-like motion to propel the cell forward. Now in humans, gentlemen, you have these, because every sperm has a flagella, because it must use it to swim to find the egg or the ovum. Functions, structures and aid in locomotion and movement of other substances or objects. When we look at the contrast, animal cells have lysosomes, centrioles, and flagella, whereas the plants do not. But the plants have the 3 C's. It has the cell wall, the chloroplast, and the central vacuole. And that's all I have for you today. So bye now.